Immunotherapy is one of the most important fields in which significant advances has been made in the last decades in the way we treat cancer patients. It improved significantly the prognosis of many patients in several types of cancer. Due to the highly divergent and individual nature of both the immune system and the developed tumors, personal medicine provides very important input regarding the decision if and how to provide immunotherapy for cancer patients. The analysis involved in this field are, though, rather complex and involves working with large sequencing data, and in general requires bioinformatics expertise and significant type and computational resources. In the scope of this exercise, we'll try to list the main areas where genomics and personalized medicine can assist in the decision process. Elad talked about the importance of mutational burden for the probability of success of immunotherapy treatment, and accordingly for the doctor's decision whether to provide such treatment. Let's open the tumor portal maintained by the Broad Institute and go to the figure on the bottom of the page designated background mutation frequencies across cancers. This figure is in the exact format of a well-known figure from Michael Stratton's seminal Nature paper from 2013 regarding signatures of mutational process in human cancers, and demonstrates the difference between cancer types and accordingly the fact that immunotherapy is not expected to achieve the same level of success in each tumor. It also demonstrates that different tumors belonging to the same cancer type can show large variability in the mutational burden. This emphasizes the importance of personal medicine and the need to get information for the specific mutational burden for each individual case. Remember that the numbers on the y-axis in this figure are in log scale. Okay, so we understand that, but how can we get information regarding mutational burden for individual samples? For research purpose, we might use some data and tools resources in the cancer portals. Let's stay in this portal, tumor portal, and go to the upper part of the page and choose one particular cancer type, say AML. We get analysis panels for mutation data and metadata for 200 patients. The uppermost graph shows the mutation rate in each and every sample. Other tracks below show additional data, including mutations in selected genes and metadata. We can sort the samples by mutation rate. Note that for AML, the mean mutation rate expressed as mutations per megabase is around 0.2. Let's open now another tumor type, say colorectal cancer. Let's sort again by the mutation rate. We can see that the average mutation rate in these samples is much higher than in AML. We can see also huge variability between the samples. Okay. After sorting the samples by mutation rate, it is also easier to find association between mutation load and mutations in individual genes. For example, we can see that there is a very significant correlation between mutation rate and mutations in the MSH6 gene, a mismatch repair gene. In general, mutations in DNA repair pathways will give rise to high tumor mutation rate. Such tumors might be better candidates for immunotherapy. I would like to mention that it is not entirely clear how we should calculate tumor mutation burden. We can check in the entire genome only within coding regions, consider frequency of individual mutations only, and consider more carefully only specific type of mutations. We might include or not include synonymous mutations. Stop gain, for example, the most drastic loss of function event usually is actually not very relevant for the purpose of neoantigen presentation. I believe some standardization in the definition of this term is about to come. In the meantime, it is very important at least to be consistent in the calculation of mutation rates or mutation burden within the samples that we have. Okay, so we now know how to explore mutational burden for public samples. But what if we want to get mutational burden for our own samples that we sequenced? Obviously, we'll need to go through next generation pipelines which will help us call variants for our samples. Given that we have the final variance calling file, the VCF file, we can now go on and estimate mutational burden and other useful information like mutations within DNA repair genes. The Personalized Cancer Genome Reporter, PCGR, is a new standalone software package for functional annotation and translation of individual cancer genomes and for precision oncology, developed in the Oslo University Hospital. The results include very useful information regarding the tumor sample, including the estimation of mutational burden and how it is compared to a variety of public tumor samples. To run such analysis, we will need to install and run the software locally. We can always turn to simpler and more basic tools which can run on the web. One of those is the web version of ANNOVAR, a highly popular platform for variant annotations called wANNOVAR. Let's get started. We should provide our non-commercial web address and sample identifier and choose our VCF file. After submitting, ANNOVAR is annotating our variants, a procedure which might take some time. When the results are ready, we get exome summary results and genome summary results. We can download these files and use them later, or continue to work in the ANNOVAR site. For now, let's open the exome summary results, as coding variants, which are the important variants for immunotherapy, can be found here. So we'll press the View button. We can see all exonic variants in our file. Below the variants, we have now sophisticated filtering menus, which allow us to filter the variants even further. Assuming that our VCF file already included only somatic variants, we can simply choose all variants which cause change in the protein sequence. We'll filter out synonymous and stop-gain mutations, as this type of mutation do not change the protein sequence and therefore cannot give rise to neoantigens. Note that if our original VCF included only tumor variants, many of these variants are not somatic necessarily or unique to the tumor, and it will be useful to exclude variants which are frequent in the general populations, as such variants most likely represent germline events. After which we can go on and filtering the mutations. The resulting list includes only coding changing variants which might be considered as neoantigens. The number of these events might represent the mutation rate for the given tumor. It turns out that knowing the tumor somatic mutations which change polypeptide sequences is just the first step for predicting if these peptides might serve as good neoantigens. When the peptides are presented on the HLA molecules, their binding affinity is strongly dependent on the sequence of the HLA molecules of the host. The strength of this interaction can be predicted, but we will need first to know the HLA type of the patient. HLA typing can be traditionally determined by antibodies screened on a blood sample or by PCR, but can also be determined based on the DNA or RNA sequencing of the patient if the sequencing data is available for us. There are today several software packages to predict the HLA subtypes in reasonable accuracy, which basically involve realignment of the whole sequencing reads to a local reference which includes the sequences of all known HLA alleles. One example of such tool is FLAT. Most of these tools require local installation of software and bioinformatics expertise. They can exploit both DNA sequencing, preferably exome sequencing, as well as RNA sequencing for the purpose of LHA typing. Given that we now know the sequences of the mutated peptides and the HLA types of the patient, we are basically ready to predict potential neoantigens for the tumor. Several bioinformatics packages and server for this task are available. An example for such tool is NetMHSpan, a Danish tool which is based on machine learning techniques. The input includes the peptide sequences and the HLA type. Let's select the peptides of length 9 and HLA alleles HLA A0101 and HLA A0201. Following submission, the program calculates the binding scores, which reflect how efficient the peptide is expected to be in activation of cytotoxic T cells. We can see that one of the peptides can create weak binding in this case. The program can be used for prediction of thousands of peptides simultaneously. We should remember that in order to efficiently calculate which mutated peptide will be an effective neoantigen, we should consider not only the antigenic potential of the new peptide sequence, but also the abundance of that peptide in the cells. For that, we need to take into account the expression level of the gene in the relevant tissue. For research purposes, we can again use the data available in the cancer portals we saw before. The GDAC Firehouse of the Broad Institute includes, alongside the mutations found in the tumor, additional molecular data such as expression level of each gene in the tumor sample. Let's learn how to obtain this data. Let's go to the GDAC website and select the cancer type of interest. For example, let's select data of colorectal cancer. Here we can find all relevant molecular data collected for patients of colorectal cancer. We can download here the list of variants as well as list with expression levels for each gene in each sample. We can then intersect the data using offline tools in order to get the expression level of each mutated gene. How do we get expression data for our own cases? Well, the best, of course, is to conduct our expression experiment abundance assay for the tumor samples, such as RNA-seq or microarrays. In the absence of such assay, we might use expression data from resources like GDAC Firehouse, or even based on resources which provide expression data for normal tissues, such as the GTEx project maintained by the NIH Consortium. To get list of gene expression in many non-op samples, we can go to the portal, shown here, and get the data from the Datasets tab, Data Download option. The data, which is available to download here, include expression level in many different tissues, which can be used to estimate expression levels in many specific tumors. So as you can see, genomic aspects of immunotherapy are still young and emerging. The tools are not yet very mature, but it is important to understand the concept and the basic steps of the analysis.